Hello, my name is Jeremy Pollock, and I'm a senior at the University of Michigan studying engineering physics and minoring in biology. So right now, I'm going to be taking you through my winter 2020 honors capstone project on the stability of B-TRCP1 under the inhibition of ATM kinase. So I did this research under the supervision of Dipankar Ray, who is an associate professor in the radiation oncology department of Michigan Medicine, and I've worked in his lab for 2 years doing research on cancer biology, specifically focused on radiation biology and various cancer treatments. So now I'll take you through the background or some background and the purpose of my project. So the purpose of this project was to better understand the stability of beta-TRCP1. So beta-TRCP1 is an E3 ubiquitin ligase, which is responsible for ubiquitinating various other proteins, primarily proteins that are involved in cell-to-cell signaling, cell-cell recognition, and cell division. So the reason why we find this important is because in the past, the Ray lab found that beta-TRCP1 plays a role in ubiquitinating KRAS, which is an oncogene that is found mutated in 30% of human cancers overall and 90% of pancreatic cancers. And these types of cancers have really high mortality rates, and to date, there are few available treatments for KRAS. So our goal is that by better coming to understand beta-TRCP1, we're hoping that we can help identify potential treatments for KRAS mutant cancers. And so, based on some of our past experiments and hypothesis that we have about beta-TRCP1 structures and the way it interacts with other proteins has made us believe that ATM kinase is a potential protein that may play a role in phosphorylating and stabilizing beta-TRCP1. So for this project, we want to take a look at that specific relationship and see if by inhibiting ATM kinase, we would be able to affect the level of beta-TRCP1 and thus identify some form of relationship there. So the experimental setup for this project was, so first of all, I cultured cells from the H441 cell line, which are a cell line that is derived from adenocarcinoma cells from a human lung cancer patient. And they have been found to have a mutant KRAS genotype, which made them the perfect model for this experiment. And then I treated the cells with various concentrations of PI103 and Ku55933, which are both ATM inhibitors. PI103 has some broader targets, while Ku55933 is just a specific ATM kinase inhibitor. And so after treatment, 48 hours later, I isolated protein from these cells and then conducted immunoblotting, which is also known as Western blotting, to quantify relative protein levels within these cells after these treatments. And the following proteins were quantified using ImageJ software. So beta-TRCP1, GAPDH, HSC70, and phospho-AKT. So now this slide here shows the results of my Western blotting for this project. As you can see from left to right, we start with the control sample in the far right, and then one treatment sample with 5 micromolars of PI103, and then 3 treatment samples with increasing concentrations of Ku55933. So in the red box here, in the top band, you can see that there appears to be decreasing levels of beta-TRCP1 across increasing concentrations of Ku55933. And we did another band that is not shown here is phospho-AKT, which was completely knocked down in all of the bands other than the control band, which showed that ATM kinase was definitely effectively inhibited in this experiment, which shows that there must be a relationship, or at least indicates that there's a relationship between beta-TRCP1 and ATM kinase. And another thing I wanted to mention is that in the second band, we see that the concentration of beta-TRCP1 after 5 micromolar treatment of PI103 is relatively unchanged. In past experiments done by the Ray Lab and by myself, we found that B-TRCP1 is normally effectively knocked down by PI103, so this result was somewhat unexpected. And because of that, we're probably going to look back at the experiment, and we will have to repeat it to gain more clear understandings of this data and to see if this experiment is effectively repeatable. But it was an interesting result, seeing that we did see that ATM kinase had been inhibited. So that's something that we'll need further study. And then here is a continuation of the results on the past page. So below is the effective relative levels of B-TRCP1 under the treatments of various Ku55933 concentrations. So for 3 micromolars, there was a 0.75 relative level of B-TRCP1 to the control when using HSC70 as the control protein for normalization of protein levels, and then 0.88 for 10 micromolar and 0.41 for 30 micromolar. So there is an effective decrease in all samples. It's a bit odd that there is an increased level in the 10 micromolar sample versus the 3 micromolar sample, but this is likely due to some of my handling, as image J measures the relative quantity based on the shape of the Western blot, and because of this, my handling may have affected the given shape of the B-TRCP1 protein band, as shown on this slide here. There may have been some effect in that fourth lane, thus changing the overall concentration or overall red concentration. So some further analysis to this is, so B-TRCP1 levels decrease with increasing Ku55933, which was something that was shown. And this goes to show that there is a potential relationship between Ku55933's target ATM kinase and B-TRCP1. And also, as mentioned before, there was some inconsistency with this trend, which was likely due to my own handling. So this experiment will have to be repeated in the future. And so some follow-up questions to this are, how does B-TRCP1 stability change with increasing concentrations of Ku55933? So in future experiments, this experiment will be ran with greater increased levels of Ku55933 to see if the level of inhibition increases. And then also we want to figure out what the specific molecular interactions are between ATM kinase and B-TRCP1 so that we can know the true biochemical details behind this interaction, which will thus help in future studies and for potential treatments. And then also we want to study what other kinase inhibitors affect B-TRCP1 stability, which will help to identify other kinases or potential interactions with B-TRCP1. And then also, additionally, as mentioned before, PI103 showed little effect on B-TRCP1 levels, which was not something that we'd expected, as this is usually used as a positive control, as we've seen the relationship in the past. So we will likely have to include these. So when we repeat these experiments, we will have to take a look at that once again. And so lastly, I just want to talk about some obstacles, overall outcomes, and the next steps for my research. So first of all, I just want to mention that COVID-19 played a role in inhibiting my ability to continue with experiments. My research was cut off at about the 9-week mark of a project that was meant to be 14 weeks, and because of that, I wasn't able to repeat some experiments that I would have liked to. So I would have liked to be able to present some more cleaner data and have more information for this presentation. But in the future, these experiments will be repeated, and hopefully all this information will be made more clear, and that will help to provide further support for the potential relationship between ATM kinase and B-TRCP1 that was identified through this research. And also, something else is just really important overall. Throughout this project, I felt my scientific reasoning skills and understanding of biochemistry was increasing and growing, and I feel like I've gained a better understanding of various proteins, molecular interactions between them, and I've become better at the lab techniques of immunoblotting and cell culturing. And so, yeah, this was a pretty exciting project. It was cool to be able to identify a potential relationship between two proteins that may have some future effectiveness in deriving clinical treatments. And I hope to use these skills that I've gained in my future career as a medical professional. So thank you for watching.